Hello everyone, Ramya, Assistant Professor of CSE Department from Rohini College of Engineering and Technology, Subject Code OMD 551, Unit 1, Topic Types of Electrodes, Surface Electrodes, Needle Electrodes, and Microelectrodes. Objective to learn about the concepts of different types of electrodes. Electrodes are employed to pick up the electrical signals of the body. In presenting a bioelectric event to the preamplifier of signal processing circuit, it is a pair of electrodes that plays the role of transducers. For example, since the electrodes are transferring the bioelectric event to the input of the amplifier, then the amplifier should be designed such that it accommodates the characteristics of electrode. Here the types of electrodes to be used depends upon the anatomical location of bioelectric event and the dimensions of the bioelectric generator. Next we are going to see the half cell potential or electrode potential. Here the voltage depends on at an electrode electrolyte interface is designed as an half cell potential or electrode potential. Here a metal solution interface and electrode potential results from the difference in rates between two opposing processes. Here the electrodes in which no net transfer of charge occurs, the metal electrolyte interfaces are called as perfectly polarized electrodes. In this, the electrodes are unhindered exchange of charges possible across the metal electrolyte interfaces are called perfectly non-polarizable. In many instances, the presence of an electrode, the potential would not be objectionable if it were stable. For example, here the figure shows the electrical equivalent circuit of a surface electrode when it is in contact with the body surface. That means it is an interface resembles a voltage source having half cell potential, that HC. It is developed due to the charge gradient and capacitor CD in parallel with a leakage resistance RD. In the half cell potential developed can be expressed by an nearest equation, that means R represent gas constant, T absolute temperature in Kelvin, F number of energy transferred or Faraday constant, N velocity of the ion. Here C1, C2 represent the concentration of the selected ion, F1, F2 activity coefficient of the ion on the two sides of the membrane, N represents the velocity of the ion. Next we are going to see the different types of electrodes. In that first one is surface electrodes. Here the surface electrodes are generally large area surface electrodes are used to sense ECG potentials. Smaller area surface electrodes are used to sense EEG and EMG potentials. It is used to measure the potential available from the surface of the skin, and it is used to sense the potential from heart, brain and nerves. In the surface electrodes we have different types. First one is metal plate electrode. Here the metal plate electrode, the size of the metal plate electrode rectangular 3.5 cm to 5 cm and circular 4.5 cm plates from German silver, nickel or nickel plates. When these electrodes are applied on the skin with electrode plate, typical DC resistance values in the range of 2 to 10 kilo ohms. The high frequency impedance amounts to a few hundred ohms. The next one is suction cup electrode. It is more practical and is well suited for attachment to flat surface of the body. This electrode has a small area because only the rim is in contact with the skin. Next type is adhesive tape electrode. It is the pressure of the surface electrode against the skin may sequence the electrode paste out. You have to avoid this problem. Adhesive tape electrode is used. It consists of a lightweight metallic screen. It is backed by a pair of electrodes. And the next one is multi-point electrodes. The multi-point electrode is a practical electrode for ECG measurements and it contains nearly 1000 fine active contact points. The next one is floating electrodes. The metal does not contact with the subject directly. It is the contact is made through an electrolyte bridge. That means by means of this electrode movement, artifact is eliminated. This is called as liquid junction electrode. The last one is Priggled disposable electrode. That means it is a disposable electrodes with the adhesive already in place or now being used regularly. Since these devices are ready to be applied to the patient, saving a a considerable amount of time. Next one is needle electrode. It is used to record the peripheral name action potential. The needle records for EMG consist merely of fine insulator wires. Their tips are in contact with the nerve, muscle or other tissue from which the measurement is made. Here the remainder of the wire is converted to some form of insulation to prevent shorting. There are two types of electrodes. First one is unipolar electrode. That means it is a single wire inside the needle serves as a unipolar electrode. And the next one is bipolar electrode. If two wires are placed inside the needle, the measurement is called bipolar and it provides a very localized measurement between the two wire tips. And the next one is microelectrodes. It is used to measure bioelectric potential near or within a single. It consists of two types, metal electrodes and micropipet. First one is metal electrodes. It is formed by electrolytically etching the tip of a fine tungsten or stainless steel wire to the desired size. Here then the wire is coated with an insulating material. Then the next one is micropipet electrode. It is a glass micropipet with the tip drawn out to the desired size. Here it is filled with an electrolyte compatible with a cellular fluids. Here we have dual interface. One interface consists of a metal wire in contact with the electrolyte solution inside the micropipet, while the other interface is in between the electrolyte inside the pipette and the fluid or outside the cell. Here in this, the metal electrode interface is between the metal film and the electrolyte of the cell. Thank you.